Okay, good evening, everyone. This side, Dr. Abhijeet. So, yes, good evening, everyone. This side, Dr. Abhijeet, and today we are going to discuss the Carbohydrate Chemistry 1, right? So, this program is being initiated by the Oracle, that is 21 days revision series live class for the university exams. Already, I have done two sessions, and today is the third session where I will be discussing the entire carbohydrate chemistry with you guys. I'm going to revise the entire topic, guys, in like Q&A form. That is the question and answer format, right? I will be telling you how to write the answer, how to approach to the question in the university exams, right? How much to write and what important points are to be included in the question, right? Which you cannot like miss out. I will be revising all the important questions, right? All the questions that I believe are potent for your upcoming no prof exam, or they have been asked in various university exams across India, right? So I will be revising it in a very crisp, conceptual, and concise manner, right? Now, guys, remember the topic that I'm discussing today, it is carbohydrate chemistry 1. So I will be discussing the classification of carbohydrates, including the mono, di, and polysaccharides, right? I will be discussing the importance of monosaccharides, the role of disaccharides in diet and digestion, the structure and function of starch and glycogen. I will be discussing like the short answers. Yeah, the difference between the alpha and the beta glycosidic bonds. What is reducing sugar and how what test no you might have done also in your practicals, right? That test I will be discussing with you guys. And at last, I will be discussing the significance of dietary fiber for health. Now, long essay questions, beta, you already know it is approximately 10 marks of questions, right? Short essays are nearly about no, they either ask it for 8 marks or 5 marks. And short answers are basically like 3 or 2 marks. Now, why to choose Oracle, guys? The very first reason is for the flexible learning, guys. You can always go on for the recorded versions. If you're not attending the classes live, you can always go on for the recorded ones that are available on the app. Even the live classes that we are conducting daily between 6 p.m. to 10 p.m. You can also go on for these recorded versions. I mean, the live classes that we are taking. The practical sessions, guys, I already know many of the students tend to ask to know they don't find a very good source where they can know actually learn the practicals. So yes, we have the recorded version of the practical sessions along with the notes also. Exam module just a month before, you guys start asking, "Okay, we don't have time to read the entire content of the book and everything." So under 20 hours, we have covered the each and important topic of the biochemistry, which I feel like it's very important for the university exams. You can always go on for these exam modules. And the one that is the hard copy book set, guys. Anywhere in India, I know you can order these hard copy book sets, which are actually containing the notes, which are actually having Q&A, that is the question and answer format. We have fun zones, riddles, MCQs, and all the topic is divided into several questions, no in long questions also, short questions also, right? So you can go on for this as well. Foundational class, guys, I've already told you for flexible learning, you can always go on for the recorded versions. The live classes that we are conducting daily from 6 p.m. to 10 p.m. The express exam module in which we are covering the entire syllabus under 20 hours. One-to-one doubt clearing session. This is a unique feature of this particular app, guys. You can always book an appointment prior, right? And you can always discuss your doubts with the respected faculty in any of the particular subject. Hard copies book sets, guys, you can always order the hard copy book sets which are now available at your doorstep. Even the practical sessions, we have the practical book also, which yeah, covering the entire no first prof anatomy, physio, and biochem. Question bank, guys, basically consists of the questions starting from basic to advanced level, right? These questions are being curated by the expert team. Along with that, we have taken questions from the previous FMG, NEET PG, and INI-CET, all the national exams, right? And nowadays, the hot favorite of the examiner, that is the clinical vignettes. They are also being curated by our expert team, keeping in view the current trend and pattern, right? So you can, after each and every topic, these question banks are there where you can go and like solve and test your knowledge. Practical sessions we've already spoken about. The subscription plan, guys, the foundational plan. We have the advanced learning plan, we have the complete learning plan, and we have the ultimate MBBS plan. Apart from that, if you want additional discounts, guys, you can go on for my code that is Dr. Abhijeet 20 for additional discounts. Now, starting with the very first question that is of 10 marks. Now, the question says, provide a detailed description of the structure and classification of carbohydrates, including mono, di, and polysaccharides. Now, guys, now let us start writing this question. Now, answer number one, that is carbohydrates. Now, carbohydrates basically means hydrates of carbon. These are nothing but hydrates of carbon. General formula. Now, for monosaccharides, guys, you already know. Monosaccharides, that is CnH2ON. Now, N basically represents the number of carbon atoms. The number of carbon atoms. One more definition that you can write, that carbohydrates are aldehyde. Now, when I say aldehyde, guys, I'm basically referring to the functional group you already know, that is CHO. And I can write CHO like this also, right? Or keto derivative. Now, when I say keto derivative, guys, you already know I'm talking C double bond O of polyhydroxy alcohol. Of polyhydroxy alcohol. They are aldehyde or keto derivative of polyhydroxy alcohol. Now, guys, when I say polyhydroxy alcohol, let me just try to what has happened. Yeah. Now, when I say polyhydroxy alcohol. Now, guys, you already know hydroxy means OH. Poly means many, right? So remember, we are taking 3 carbons that are bonded to the OH group, right? And they're bonded to each other. Now you already know carbon forms, yes, 4 valencies are there, right? So it forms 4 bonds. Just to balance that, I will put hydrogen. Yes, the first carbon, 1 bond with OH, 1 bond with another carbon, and 2 hydrogen bonds. Second carbon, 1 above, 1 with OH, 1 with H, and 1 with the down carbon, the third carbon. Right? So it will be like this, 1, 2, and 3. Now this structure, it is basically of shit. Yes, you're right, guys. This is the structure of the glycerol. Glycerol. Now, carbohydrates are aldehyde or keto derivative of polyhydroxy alcohol. Now, when I say polyhydroxy alcohol, guys, let me just try to see this over here. Yeah, we have yes. Now for aldehyde group, guys, for aldehyde group, what I'm doing, I'm removing this H2 over here. One hydrogen from here and one hydrogen from here, from the first carbon. Obviously, carbon will be left and it will be bonded to hydrogen, and only one hydrogen is left on the other side. The other entire structure remains the same, minus H2, right? So this structure, guys, this structure, it is nothing but glycerol. All alcohol is changing into aldehyde, so it is called as glyceraldehyde. Glyceraldehyde. Remember, guys, this CHO, it is nothing but the aldehyde derivative, right? Aldehyde derivative is always present at first carbon, right? And this glyceraldehyde is nothing but the parent carbohydrate. The parent carbohydrate. So you can see that, again, justifying my definition, carbohydrates are aldehyde groups of polyhydroxy alcohol, right? On the other hand, they can be either keto derivatives also. Keto derivatives. Now, what I'm doing for the keto derivative, guys, I'm removing hydrogen from the second carbon, right? Keto derivative is always present at second carbon, not at first. Aldehyde, it is always present at the first carbon, right? Now, what I'm doing, yes, we have COH. Again, the entire structure will remain the same. And when I'm removing both the hydrogen, it is C double bond O, it is COH, and it is H2. This structure, guys, if you just see over here, this is the acetone. CH, C double bond O, CH2. I mean, yes, just let me. CH2, C double bond O, CH2. The structure is acetone, and two hydroxy groups are present at first and third carbon. So I can always write this. It is called as dihydroxyacetone. Dihydroxyacetone. And remember, it is also a carbohydrate. Carbohydrate. Glyceraldehyde, it is the parent carbohydrate. Dihydroxyacetone, it is also a carbohydrate. So yes, you guys already know that this structure. So again, justifying my definition, the carbohydrates are aldehyde or keto derivative of polyhydroxy alcohol. Alcohol over here, which I have taken, it is glycerol. It is glycerol, right? Now coming on to the classification of the carbohydrates, right? So again, guys, the questions that they always ask, the parent carbohydrate and dihydroxyacetone, the glyceraldehyde and this are nothing but the simplest carbohydrate. They are nothing but the simplest carbohydrate, right? And if I ask you what is the parent alcohol from which the carbohydrates are being formed, the parent alcohol is nothing but the glycerol. Nothing but the glycerol. Now coming on to the classification of carbohydrates, guys, you already know, if we talk about the classification of carbohydrates. Now, again, just try to make a flowchart, guys. Always in this answer, if you can make a flowchart, that will be like more good. The first one, remember, it is monosaccharide. The second one, polysaccharide. Sorry, let it be like this first. Second one, disaccharide. Then third one, oligosaccharide. And the fourth one, polysaccharide. Fourth one, polysaccharide. Now, when I say monosaccharide, mono means single. Single saccharide means sugar. Single sugar unit. Disaccharide means two potential sugar units. Oligosaccharide means few sugar units. And basically, when I say few sugar units, I'm actually referring to the carbon number. I mean, the 3 to 10 sugar units, actually. Polysaccharides more than 10 sugar units. Now, coming on to the monosaccharides guys first. Monosaccharides, remember the sugars which cannot be hydrolyzed further. We cannot break these sugars no, by utilization of water again. These are the simplest sugars, right? Which cannot be hydrolyzed further. It contains one single sugar unit. And they are building blocks of all carbohydrates. They are building blocks of all carbohydrates, right? Now, if we talk about the monosaccharides, I'm just discussing the monosaccharides in detail, guys. You just have to draw one more table. Now, depending upon the number of carbon atoms, okay, let us take number of carbon atoms over here. Now, what will be the general name? And we have already discussed that we have two forms, either aldoses or ketosis. Carbohydrates, either they are containing the CHO group, that is the aldehyde group, or the C double bond O, that is the keto group. Now, let us start from the very first one, that is the number of carbon atoms, let it be 3. Now, guys, the general name it is called as triosis. The general name it is called as triosis. And you already know we have just made the structure right now. We have just drawn the structure of this carbohydrate triosis. You can see 3 carbon atoms over here, guys. I hope you can very well appreciate we have these 3 carbon atoms. And aldehyde group, it is called as glyceraldehyde group. And for keto group, it is called as dihydroxyacetone. So for 3 carbon atoms, guys, for 3 carbon atoms, yes, it is glyceraldehyde. And ketosis, it is dihydroxyacetone. 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 The fourth one, we have tetrosis. Okay, the aldehyde derivative, it is called as erythrose. And keto derivative, it is called as erythrulose. Erythrulose, right? The fifth one, we have pentosis. Pentoses again occur in 3 forms, right? Depending on the orientation of H and OH, right? We have ribose, xylose, and arabinose. For ribose, we have ribulose as a keto derivative. Xylose, we have xylulose. And for arabinose, we have nothing. Right? You can all draw these structures and no practice on. For 6, we already know the hexoses. Now, hexoses, again, they will exist in some forms. We have glucose, galactose, and mannose. These 3 are nothing but aldohexoses. Aldohexoses. In MCQ also, they will now ask you a question. All of the following are examples of aldohexoses except. So remember, we have 3, glucose, galactose, and mannose. Hexoses means they are containing how many carbon atoms? Status 6. How many carbon atoms? Status 6. Remember, for glucose only we have that is called as fructose, and fructose is called as a ketohexose. fructose, it is called as the ketohexose, right? So remember, guys, this is something that you have to remember. Over here, one more thing that I wanted to assure you, that is, yes, the ribose, okay, or if we talk about the pentose especially, right? The ribose and the deoxyribose, they all constitute a part of nucleic acid. It constitutes a part of nucleic acid. Again, one important no importance of pentoses. Like we have, I have a table also. Let me just try to, yes, this particular table which I've already taken from the Auricle notes. You can just now glyceraldehyde and dihydroxyacetone. They are nothing but they are the parts of glycolysis. That is nothing but the carbohydrate metabolism, right? Intermediate in glycolysis, breakdown of glucose, okay, into pyruvate. You will be learning this in the metabolism part in the next one, right? So just remember. Tetrosis, it is again an intermediate, right? It's 4-phosphate. Now, when I say 4-phosphate, guys, I'm taking 4 carbons, and to the fourth carbon, I'm adding phosphate group, right? So erythrose 4-phosphate, it's an intermediate in carbohydrate metabolism, like learning later on. Ribose, deoxyribose, they all are actually no important components of the nucleic acids. Even this ATP, adenosine triphosphate, or NAD, NADP, right? Glucose, you already know it is the sugar fuel of our life, right? So yes, that is you have to remember. Okay, so these are the basic ones which you can remember according to the importance and know the structure and classification of monosaccharides as per concerned. Now coming on to the next one, that is the disaccharides. The disaccharides, yes. Now disaccharides are nothing but they are actually they have asked all the three, the mono, di, and polysaccharides. So remember, guys, the disaccharides, yes. Two monosaccharides are basically joined by glycosidic bond. Now, when I say glycosidic bond, it is called as the covalent bond. It is called as the covalent bond, right? Now, this glycosidic bond, it is basically no dividing it into reducing and non-reducing sugar. Reducing and non-reducing sugar. Now, guys, again, there will be a question how to identify which sugar is reducing and which sugar is not reducing. Now we have different tests also, but according just by watching the structure, okay, remember that when I say reducing sugar, remember at least one functional group should be present. One functional group should be present. One free functional group. Remember, guys, free word is very important. One free functional group should be there. Okay, either it could be a aldehyde group or either it could be a ketone group. Right? The functional group, the free functional group is the one which makes it reducing in nature. Patish, just a moment. Sorry for the interruption. Okay. Now, when I say the non-reducing disaccharide, remember the two functional groups, okay, are joined. Now, when the two functional groups are joined, again, there will be no free functional group. No free functional group. Okay, because both the functional groups are engaged into the linkage. So obviously that there will be no functional group, right? No free functional group will be there. Let us take a very common example over here. I have to explain this in other question as well, but let us just try to understand it over here only. We have 6 carbons, okay? And you already know, yes, third carbon H is over here. I mean, OH is over here, H will be here, right? So remember guys, this is one glucose. This is one glucose, right? And let us just try to take another glucose as well. The same structure. Now both the structure is of glucose. Now when I say disaccharide guys, disaccharide means basically two monosaccharides are joined together by glycosidic bond, right? Now we have the functional group aldehyde group at the first carbon of the first glucose. And remember guys, whenever the first carbon atom of the first glucose will be joining together with the fourth carbon atom of another glucose. Okay. Now this structure it is called as maltose. Maltose it is formed from glucose plus glucose and it will be alpha 1 to 4 linkage. Alpha basically means the carbon atom of the first glucose, which is actually a functional group, will be joining to the fourth carbon atom of another glucose. They both will be joining together by alpha 1 to 4 linkage. Now why it is reducing? Now it says that it's a reducing sugar. Now why it is reducing guys? Remember that at least one free functional group should be there. Now see if the entire structure is of maltose now, you can very well see in the second glucose, the first carbon is aldehyde and it is free. It is not participating in the linkage. Again, it is not participating in the linkage. That's why it is the free functional group which makes it reducing. The free functional group which makes it reducing, right? So yes, now we have done this reducing. Okay, isomaltose, glucose plus glucose again. The first carbon atom and it is bonded towards the sixth carbon atom. Again reducing guys because again the free functional group is present. The other glucose its free functional group is not utilized. Trehalose guys or sucrose. Sucrose is glucose plus fructose. Now I have already told you it's alpha 1 to 2 linkage. One carbon, the first carbon atom of the glucose, it contains the aldehyde group. The second carbon atom of the fructose contains keto group, and both of them are participating into the linkage. It means that both the functional groups are engaged into the linkage, leaving no free functional group. That's why it is called as the non-reducing sugar. That's why it is called as the non-reducing sugar. And the general formula of disaccharides it is CnH2n minus 1. CnH2n minus 1, right? And represents the number of the carbon atom, right? So that is maltose, sucrose, lactose is galactose plus glucose. We have beta 1 to 4 linkage guys. Now you already know galactose also will contain CHO group. Glucose also will contain CHO group. The difference is in the orientation of H and OH. Now over here guys, remember in galactose and glucose, what is changed guys? What is changed? The fourth carbon atom will be having OH group over here and H over here. From glucose, how galactose differentiates, both of them are having the aldehyde group, correct? In glucose, we have at the third carbon, OH is on the right side. In galactose also, in galactose also, we have this OH on, yeah, this left side, right? But at fourth place also, OH is on the left hand side, right? So that is the change. And you can very well see when glucose and galactose are combined, right? We have two different structures. One is galactose, one is glucose. That's why it is beta 1 to 4 linkage, 1 to 4 linkage, right? The first carbon atom of the glucose will be bonded towards the fourth carbon atom. Sorry. Okay, I have taken it wrong. We have to take opposite ways, right? So yes. The first carbon atom of the galactose will be bonded to the fourth carbon atom of the glucose, right? So we are having a free functional group in the glucose. Now this free functional group, it is not participating in the bonding. That's why it is called as reducing sugar. That's why it is called as reducing sugar, right? So remember, yeah, galactose and glucose. Yes, galactose and glucose are different compounds. That's why beta. 1 to 4, 1 means the carbon atom at the galactose will be bonded. The carbon atom at the galactose, it is basically, yeah, it is containing the aldehyde group, right? It is containing the aldehyde group and it is bonded to the fourth one, okay, of the glucose, beta 1 to 4, right? So yes, that's why it's a reducing sugar. Now, the sucrose, guys, it is a common commercial product. Okay, and you already know it is consisting of glucose and fructose. And remember, guys, these are the basic things that you already know, right? Glucose and fructose. The glucose structure you already know. If suppose this is a glucose containing 6 carbon atoms, and you already know the aldehyde group, it is present at the first carbon. This is glucose. And for, yes, the fructose, you already know, we again have 6 carbons, but it is a ketohexose. So it will be having the keto group at the second carbon. So the first carbon, it is bonded to the second carbon of fructose. And basically, when both the functional groups are engaged into the linkage, there is no free functional group. That's why it is the sucrose, it is a non-reducing sugar. It is a non-reducing sugar. It is a non-reducing sugar, right? It's a non-reducing sugar. Now, it is sweeter than most common sugars such as glucose, lactose, and maltose, except for fructose. In intestine, the enzyme sucrase, remember guys, the enzyme sucrase hydrolyzes sucrose into glucose and fructose. Then they are absorbed into the body. Then they are absorbed into the body. Maltose is basically a disaccharide from 2 units of glucose. It is called as malt sugar, right? Source for growing embryo in the seed, and it is also used in alcohol production. Lactose, guys, you already know it is commonly called as the milk salt, okay? And lactose, it basically derived from the condensation of galactose and glucose, right? So the enzyme lactose will be digested by, the disaccharide lactose will be digested by an enzyme called as lactase, and it's also used as a filler in tablets, and it is widely used in food industry. Now that was regarding the, yes, the disaccharide. So the question says we have already covered, yeah, the monosaccharides and the disaccharides. Now coming on to the polysaccharides, guys, coming on to the polysaccharides. Now in polysaccharides, guys, the little bit of information that I wanted to share with you people. Polysaccharides, it is more than 10 sugar units, and they are divided into homopolysaccharide and heteropolysaccharide. Now, homo means over here it is homoglycans. Homoglycans. Homoglycans means basically one type of sugar or monomer units. One type of sugar or monomer units. Heteropolysaccharides, also called as heteroglycans. These are more than one type of monomer units. More than one type of monomer units. Okay, more than one type of monomer units. Again, the examples are very, very important over here. Examples over here, homopolysaccharide, it is glycogen. We have, yes, starch, cellulose, inulin, chitin. Heteropolysaccharides, you can just remember example. We have already discussed this topic earlier also. Glycosaminoglycans that are called as GAGs. We have agarose, we have pectin. These are all examples of heteropolysaccharides. So that is all about the very first question that we have discussed. Yes. So yes, the classification of carbohydrates, structures are monosaccharides, disaccharides, and polysaccharides. Coming on to the next question, guys. Again, this question has been taken from one of the previous year questions. Describe the importance of monosaccharides in energy metabolism. Okay, excluding the detailed metabolic pathways. Metabolic pathways are the ones which we will be discussing in the next the next semester. So, guys, the very first one that you have to remember over here, we are talking about the glucose. Glucose is one of the primary energy source, okay, for cells, right? Now, glucose is basically metabolized. It is metabolized to form this, yes, to produce ATP. ATP is nothing but the energy currency of the cell. Energy currency of the cell. That is, yes, the energy currency of the cell. That is, you already know triphosphate. Adenosine triphosphate, right? Adenosine triphosphate, right? So ATP is the one which is required for all cellular activities, even for muscular contractions, right? Protein synthesis, transportation, transport across cell membrane, and many, many other things, right? So remember, guys, glucose, it is also used as primary fuel, okay, by the very important structure of the brain, that is, of our body, that is the brain, right? So remember, yes, yeah. Hmm. Even ribose, okay, ribose also, you guys, you already know it's an important component of nucleotides. Okay, it's an important component of nucleotides. Nucleotides are the building blocks of RNA and DNA, right? So remember these RNA and DNA. Basically, they are essential for cellular activities. They are essential for cellular activities, right? Apart from that, guys, yes, we have cellular signaling also, right? We have various other processes. For example, the regulation of blood sugar levels, right? So they are also being known, taken care of, right? So yes, even the blood sugar levels. blood sugar levels when there is a fed state and the fasting state. Fed or the fasting state. Fed state means just you have consumed the food nearly like 1 to 4 hours after food. So what the body will do, body will try to take up the glucose and will utilize it for various processes like for generating the ATP for various metabolic processes. But the extra glucose, for example, you drink 1 liter of Coca-Cola. Yes, so majority of them will be taken care by the liver. The glucose will be absorbed into the stomach and then it will go into the liver where the glucose will combine together and form glycogen. So just after 4 to 16 hours in which you are not consuming food, that is called as the early fasting state, that glucose which was stored as glycogen, okay, will be now broken down to give energy to the various cellular activities. Right, so that is the importance that we have to remember. Next question, explain the role of disaccharides. I think we have discussed this disaccharides in detail, guys, right? Disaccharides, yes, they are from energy sources, right? The energy sources. So you already know the disaccharides. We have maltose, isomaltose, right? So you have to remember all of them, lactose, lactulose. What are the monomer units? You already know the disaccharides. They are formed from the, yes. So remember it is glucose plus glucose. And linkage also you have to remember it is alpha 1 to 4 linkage. Glucose plus glucose, but the linkage over here it is alpha 1 to 6 linkage. Then we have lactose, that is galactose plus glucose, that is beta 1 to 4 linkage. Lactulose, it is the human one made, that is galactose plus fructose, right? So you have to remember these are all examples of reducing disaccharides. They all have the free functional group, right? The other ones which you can remember for non-reducing, the non-reducing disaccharides, which are sucrose and trehalose. Sucrose basically consists of glucose plus fructose, and trehalose consists of glucose plus glucose. Linkage, it is alpha 1 to beta 2 linkage. And trehalose, it is alpha 1 to 1 linkage. Alpha 1 to 1 linkage. Alpha 1 to 1 linkage. Right? Now, if we come to the digestion of these disaccharides, now these are the disaccharides that you know. Coming on to the digestion of disaccharides. We've already discussed this, just try to remember, guys. Now, what is happening, guys, if you are consuming the food through the oral cavity, we are consuming disaccharides. We are consuming the table sugar, right? The disaccharides, which are nothing but the three, majorly sucrose, lactose, and maltose. Sucrose, lactose, and maltose. Now they are going inside, right? Now what will happen? The sucrose, maltose, and lactose will be broken down by the help of different enzymes. The sucrose will be broken down by the help of enzymes sucrase, maltose into maltase, and lactose into lactase. Basically, these disaccharides are broken down by the help of enzymes into monosaccharides, right? Okay, so the sucrose will give glucose plus fructose. Maltose, you already know, glucose plus glucose. And lactose will again give me, yes, glucose and galactose. Now the question is in the exam that they ask, carbohydrates are digested to form. Majority of you answer only glucose, guys. It is wrong. Carbohydrates are the end product of carbohydrate digestion. It is not glucose only. It is glucose plus fructose plus galactose. Glucose plus fructose plus galactose. Right? So that is something you have to remember over here. Now coming on to the next one. Outlining the structure and function of starch and glycogen. You already know the starch and glycogen. We just discussed that they are polysaccharides. Now, when I say polysaccharides, they have more than 10 sugar units, right? They are, yes, polysaccharides, and mostly these are nothing but the, yes. Okay, more than 10 sugar units, guys. You already know these are nothing but which class of polysaccharides? It is the homoglycans. Homoglycans or homopolysaccharides, they contain one type of sugar units, right? And the examples are also we have glycogen, starch, cellulose, inulin, chitin, right? That we wrote already. So glucose is the repeating unit in both the structures. Whether we are talking about starch, whether we are talking about glycogen, they both will have glucose as their basic structure, right? Now, they are all having alpha glycosidic bonds in their structure, which contribute to their branching patterns. Now, coming on to the structure and yes, the starch and glycogen, guys, the starch and glycogen. So remember, if we talk about the starch, it is the storage form of carbohydrates in plants, and it consists of amylose and amylopectin. Remember, guys, amylose is soluble, and remember, amylose is unbranched. One more important point that you have to remember, it is unbranched. Amylopectin, it is the one which is insoluble and it is highly branched. It is highly branched, right? So if I just try to draw amylopectin over here, it is just like this. These are the glucose units and it is like branching at different points. But if we talk about amylose, amylose is nothing but it is just like a helical structure. It's just like a linear structure like this helical one, and the glucose units are like kept like this, right? So this is the amylose and amylopectin, right? Now, if we talk about glycogen, guys, it is made up of glucose, right? It's the storage form of carbohydrates in animals, including humans. Now, what happens over here, guys? We have these glucose units. If we try to draw the structure of these glucose, I mean glycogen, so it will have these glucose units, and these glucose units are basically branching at different levels. In a linear structure, the linkage is alpha 1 to 4, but at the branching point, it is alpha 1 to 6. Only at the branching point it is alpha 1 to 6. Otherwise, rest places we have alpha 1 to 4 only, right? So remember glycogen, yes, this is the one which is written, one at branching points, right? So remember it function as a short-term energy reserves in human body, particularly in muscles and the liver. So glycogen, it is stored in two organs, guys, the livers and the muscles over here. Remember, guys, the muscles contain the glycogen in the form of beta particles, right? Beta particles, and single beta particle has 60,000 glucose units, right? So if they ask you total content of glucose, it is more in the muscle. Remember, total content of glucose is more in muscle. But if they ask you percentage by tissue volume, glycogen is more in the liver. Glycogen is more in liver, right? So rosettes of beta particle basically, in liver, basically it is stored as the aggregates of beta particles. Okay, they are highly dense, highly compact structure. right? So this is the one which you have to remember, the differences and the similarities, the structures and the functions of starch and glycogen in energy storage. Difference between alpha and yes, beta glycosidic bond, guys, have already told you it's just on the orientation, guys. It's just on the orientation, right? We have already discussed. We have already discussed this earlier as well, right? Yes. So in the alpha glycosidic bond, guys, in the alpha, yes, glycosidic bond, remember the OH group, it is attached to the anomeric carbon, right? Now, what is this anomeric carbon? It is the carbon, okay, it is the carbon derived from the carbonyl group of the monosaccharides, right? So, yes, whereas beta, it is the hydroxyl group attached to the anomeric carbon, oriented upwards when the sugar is in the ring form, right? So, yes, we have already discussed about alpha 1 to 4 glycosidic linkage, right? Whenever both the structure is of glucose, right? Glucose consists of, yes, the 6 carbon atoms. This glucose will also have the 6 carbon atoms. The first carbon of the first glucose will be bonding towards the fourth carbon atom of the second glucose, right? Both the structures are same, alpha 1 to 4 glycosidic linkage, right? Yes. And for the beta 1 guys, also I have taken beta 1 to 4 linkage, right? Cellulose, we have beta 1 to 4 glycosidic bonds, right? Right. So yes, that's why no, we people are not able to digest cellulose because the enzyme cellulase that breaks this beta 1 to 4 glycosidic bonds is basically absent, right? Reducing sugar, and I think I have defined what is the reducing sugar and how it can be identified, guys. The very first one that you have to remember, it is the Benedict's test. Benedict's test. So you already know the Benedict's reagent contains copper 2 plus ions. Benedict's reagent basically contains copper 2 plus ions, which is nothing but blue in color, right? Now, whenever a reducing sugar is present, now when I say reducing sugar, it means that there should be a free functional group, right? Now, what it will do, it will reduce this copper 2 plus ions, right, into copper 1 oxide, right? So that is nothing but just like it, or write it like this, CO2O, right? So remember, this blue color will change into orange color, or more precisely, it is red color. Okay, orange solid or red color, right? So this color change, it is indicating that this is basically a reducing sugar, right? If blue remains blue, no reducing sugar. If blue changes into orange or red, it means that there is reducing sugar. Okay. Now we have one more test for aldoses that you can remember. Yes, this is nothing but the tolerance test for aldoses, right? So tolerance test, basically the tolerance reagent, it consists of, yes, the silver nitrite in ammonia solution, right? So aldehydes, now when I say aldoses, it means aldehydes. Now aldehydes can reduce silver ions. It can reduce the silver ions to metallic silver, forming a silver mirror surface on the test tube. forming silver mirror surface on test tube, right? So if the silver mirror surface is formed, it again introduced that there is a reducing sugar with an aldehyde group, right? That is something which you can remember. Coming on for the last one for today, the significance of dietary fibers for the health. Now, guys, remember we have two types of dietary fibers. Either they are, yes, soluble, or they are insoluble dietary fibers. Now, when I say soluble, it dissolves in water to form, yeah, it dissolves in water to form a gel-like substance. And the soluble ones are basically oats, or there could be beans, or there could be lentils, or there could be apples, or it can be citrus fruits. Now, the insoluble dietary fibers are the one which do not, which is water insoluble. Okay. And it adds bulk to the stool, right? Yes. You already know the examples, the one which we are eating daily, that is the whole grains, nuts, right? And the skins of fruits and vegetables. Now, what is the important significance, guys? Remember that it helps in promoting healthy bowel movements, promoting the healthy bowel movements, right? So the insoluble one will be increasing the bulk and the weight of the stool, which helps prevent constipation by promoting regular bowel movements, right? The second important one that you can remember, it prevents diverticulosis. By improving the bowel regularity, fiber helps reduce the risk of discovering this diverticulosis. It is a condition, no, diverticulosis is a condition where there is small pouch, pouch-like structure coming from the walls of colon, coming out from walls of colon, right? And it also supports the gut. It also supports the gut microbiota. Microbiota. Right? So that is regarding the significance of dietary fiber for health. So yes, thank you everyone for your patience and for listening me out. These are the various social media handles where you can go and check on for the next schedule. Thank you.